Okay, so today we are going to discuss a very important topic as far as this respiration is concerned, and it is nothing but the mechanism of aerobic respiration. Because during yesterday's discussion, what we have said that in the availability of oxygen, aerobic respiration takes place, and in absence of oxygen, anaerobic respiration takes place. Obviously, aerobic respiration is more efficient as it produces around 38 ATPs. And anaerobic respiration, it is comparatively a little bit, we can say, less efficient as only two of them, the ATPs are available. So obviously, we have to discuss the mechanism of aerobic respiration in detail. And accordingly, we can have the basic definition as it is the stepwise breakdown of respiratory substrate. Obviously, that is glucose, and that is converted to CO2 and H2O in presence of atmospheric free oxygen. It is called as aerobic respiration. Now, it is actually a multi-step enzyme mediated energy liberating catabolic processes. So you can see too many adjective. Number one, it is multi-step because the reaction is not single step. As the energy liberation, it should not be rapid, it should not be with a boom, but it should be slow, gradual. Then every step, every reaction will be enzyme controlled, enzyme mediated. Then energy is liberated, means it is exergonic, or you can say exothermic reaction where energy is liberated, and basically organic compound glucose, it is broken down, so it is called as catabolic process. Now it involves complete oxidation of respiratory substrate glucose with oxygen, which is acting as terminal oxidant. Now what is complete oxidation and what is incomplete oxidation? Actually आपण जर का तुम्ही एखादा वूड बघितला असेल, लाकूड बघितलं असेल, ते जर आपण जाळलं तर दोन कंडिशन होऊ शकता. इफ इट इज बर्न कम्प्लिटली, इट विल बी कन्व्हर्टेड टू ऍश. अँड इफ इट इज वी कॅन से वाईल बर्निंग, आपण ते विझवलं, पाणी वगैरे टाकून दिलं, तर इट इज कन्व्हर्टेड टू कोल. बघितलंय का हे की कम्प्लीट बर्न झालं तर ऍश तयार होते. वी कॉल इट ॲज अन राख मराठीमध्ये. आणि त्याच्या उलट जर का जळताना पाणी टाकून दिलं तर त्याचा कोळसा होतो. बघितलंय का? येस ऑर नो?
हो सर.
Yes, obviously. When it is completely oxidized, उसको बोलेंगे ash. And incompletely oxidized, उसको बोलेंगे coal. Here the respiratory substrate glucose is completely oxidized, means it is converted to smallest available component CO2 and H2O. And for that oxidation, the last one, it is the oxygen which is acting as terminal oxidant, the last oxidant. And obviously, yesterday itself, we have said that the end product of this aerobic respiration, that is CO2 and water. And in addition to that, it is the process of aerobic respiration that can be completed within 4 consecutive stages. The first one, glycolysis, glucose is broken down. Glycolysis, oxidative decarboxylation. With oxidation, carbon dioxide is removed, so it is decarboxylation. Then after the name of the scientist, Krebs cycle, and it is also called as tricarboxylic acid cycle, or we can say TCA cycle. And the last step being terminal oxidation, the last oxidation where electrons and protons will be accepted by the oxygen itself. So obviously, one by one, we have to see in detail. Let's start with the discussion of first one. As I have said, glycolysis. Glyco stands for sugar, glucose, and lysis, that is breakdown. So it is obviously the first step in the respiration that occur within the cell. So it is called a cellular respiration. What it does? It brings about breakdown of glucose or related hexose sugar containing 6 carbon. It may be fructose. Like glucose and fructose are broken down into 2 molecules of pyruvic acid through enzyme mediated reaction. Now glucose, fructose, they are called as hexose. Why? They contain 6 carbon. And they are broken down in the middle. That is, they will be converted to 2 molecules containing 3 carbon each. And that 3 carbon organic acid, we call it as an pyruvic acid. So end product of glycolysis will be pyruvic acid, and that occur in a stepwise gradual reaction. That is enzyme mediated reaction, enzyme controlled reaction. The molecules of ATPs and NADPH2, they are produced, and that is very important. Some of the ATPs and NADPH2, they are the reduced coenzyme, they are produced. Now actually the entire process of glycolysis that was discovered by three German scientists. The first one, it is Amden. The second one is Meyerhoff, and the third one, Parna. So Amden, Meyerhoff, Parna. Collectively, first alphabet EMP. So this glycolysis is also described as EMP pathway. And actually, it was referred as, it was given the status as core respiration, important, central, core respiration by another scientist named James. Actually, for this entire process of glycolysis, there is no direct role of oxygen. Oxygen is not required. So you can say it is an anaerobic phase of aerobic respiration. Kindly mark my words. It is the अनएरोबिक फेज ऑफ एरोबिक रेस्पिरेशन. मींस रेस्पिरेशन इज एरोबिक, लेकिन उसका पहला स्टेप उसको ऑक्सीजन का जरूरत नहीं है, तो उसको बोलेंगे अनएरोबिक फेज ऑफ एरोबिक रेस्पिरेशन. एंड ऑब्वियसली 6 carbon glucose will be broken down to 2 molecules of pyruvic acid. So there will be no removal, there will be no release of carbon dioxide. So that is what we can say the glycolysis. Now this glycolysis, as it is anaerobic phase, it is common for both aerobic as well as anaerobic. It may be aerobic respiration, it may be anaerobic respiration. In both, the step 1 will be glycolysis. Now, in anaerobic organisms like bacteria, like yeast, there is only one process of respiration, and that is what we can say. Glycolysis is the only mechanism of respiration in anaerobic organisms like bacteria, like yeast. It occurs in the cytoplasm of the cell. Very importantly, normally we say respiration takes place within the mitochondria. भाई नहीं, respiration का पहला step cytoplasm में बनेगा, फिर दूसरा phase mitochondria में. So it is occurring, which is glycolysis is occurring within the cytoplasm of the cell. And during this process, one molecule of glucose containing 6 carbon undergo oxidation to form 2 molecules containing 3 carbon. Pyruvic acid will be produced. In case of plant, glucose is derived from sucrose, or you can say the storage carbohydrates. In case of plant, the glucose is available from sucrose, or even we can say storage carbohydrates like starch. And that sucrose, a disaccharide is converted to glucose and fructose. And that is done with the help of an enzyme. Usko bolte invertase. A fructose, a glucose is produced by breakdown of sucrose, a disaccharide, and that is with the help of enzyme invertase. Now, these two, either glucose or fructose, both of them are monosaccharide. They enter into the process of glycolysis, and then they will undergo the breakdown. Now, let's see what exact stages are there in the glycolysis. Here you can see the flow chart is there, and you know, all of us know that this is the most beautiful thing. In case of photosynthesis, मैंने बोला था, what is that? Reverse of glycolysis. आता इथे normal glycolysis है. बघा, glucose being the respiratory substrate containing 6 carbon. Now this 6 carbon glucose is inactive. It is inert. तो उसको थोड़ा जिंदा करना पड़ेगा, and then it will take part in the reaction. So glucose undergo activation by using ATP energy. ATP will be converted to ADP, and that occur in presence of magnesium, and the enzyme is hexokinase. So automatically a fructose from the ATP that contain phosphate, that phosphate will be removed and it will be given to glucose. So automatically glucose will become glucose 6-phosphate containing phosphate group at carbon number 6. And everyone know glucose is the isomer of fructose. We call it as an glucose as the bhauband of fructose. So glucose 6-phosphate will be converted to fructose 6-phosphate. Again, magnesium plus plus as well as phosphohexokinase or isomerase will be the enzyme. Now this fructose 6-phosphate, which is containing one phosphorus, phosphate group, another phosphate is added, another ATP will be used, which is converted to ADP. Mg++ rahega, phosphofructokinase will be the enzyme, and then it will contain 2-phosphate. So fructose 1,6 biphosphate will be produced. Fructose 1,6 biphosphate containing phosphate at carbon number 1 and carbon number 6. It will be produced and they will contain 6 carbon. Now due to 2-phosphate group, it become hyperactivated, and hyperactivated become unstable, and that unstable fructose 1,6 diphosphate will be broken down into two components, two parts, each containing 3 carbon. The first one is dihydroxyacetone phosphate. Dihydroxyacetone phosphate containing 3 carbon, and the other molecule will be glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate again containing 3 carbon, and again dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they are the isomer. So automatically we can say dihydroxyacetone phosphate molecule will be converted to glyceraldehyde 3-phosphate. Now there will be 2 molecules of glyceraldehyde 3-phosphate which undergo further reduction, oxidation, and during that, what happens? Number 1, it will be the glyceraldehyde phosphate dehydrogenase. Dehydrogenase that remove hydrogen, and removed hydrogen will be accepted by NAD. NAD will be converted to reduced coenzyme and ADH2. As a result of that, there will be addition of inorganic phosphate at the same time. So hydrogen removed, inorganic phosphate added, and there will be formation of 1,3 biphosphoglycerate containing 2-phosphate group at carbon number 1 and carbon number 3. That is 1,3 biphosphoglycerate. Now 1,3 diphosphoglycerate contain 2-phosphate. उसमें से एक निकाल लिया phosphate, and it is given to ADP, so it will be converted to ATP, and 1,3 biphosphate or phosphoglycerate will be converted to 3-phosphoglycerate. Now this 2-phosphoglycerate will undergo intramolecular rearrangement. बोले तो क्या? 3-phosphoglycerate में phosphate रहेगा carbon number 2 पे, sorry, carbon number 3 pe, and from that carbon number 3 the phosphate will be shifted to carbon number 2, and there will be formation of phosphoglycerate containing phosphate at carbon number 2, so it is called as 2-phosphoglycerate. Now this 2-phosphoglycerate in presence of Mg++ and another enzyme enolase, there will be removal of water molecule, and removal of water molecule, you can say dehydration reaction. 2-phosphoglycerate will undergo dehydration to form phosphoenol pyruvate. Now phosphoenol pyruvate further in presence of Mg++, potassium, and enzyme pyruvate kinase, 1-phosphate will be removed. Only 1 phosphate was there, and that will be removed, and that removed phosphate will be accepted by ADP to get converted to ATP, and as a result of that, the last product of glycolysis, that is pyruvate, or you can say pyruvic acid, will be produced. As a result, we can say at the end of glycolytic processes, what is produced? It will be the 3-carbon pyruvic acid will be produced. Are you getting this?